Pessoal, boa tarde a todos. Então, agradeço uma vez mais, estão aqui presentes assistindo esse CEBIMário de hoje. Então, hoje vamos ter a presença então da doutora Tayler. Eu quero agradecer a doutora Tayler ter aceitado participar desse evento. Então, o CEBIMário de hoje será em inglês, mas suas dúvidas podem ser escritas em português ou espanhol ou inglês para a gente, depois da apresentação da doutora Tayler, conversar um pouco mais com ela sobre os tópicos que ela vai falar hoje. Então, thanks Taylor for accepting participating in this talk. I think it is very interesting you talk about the climate change and different aspects of oxygen in the tropical Eastern Pacific. And well, I explained all, and now is your opportunity for talk. Thank you so much.
Thank you very much, Juan Pablo, for the opportunity to be sharing with all of you some of the results of actually my PhD thesis. So I'll start by telling you a little bit about who I am. My name is Tayler McLellan Clarke. I actually grew up in Costa Rica. I did my bachelor's degree at the University of Costa Rica. I studied biology there. And then I did my master's at the University of Costa Rica as well, with a more applied biology and coastal management master's program, where I worked with shrimp trawl fisheries. And after that, I took a little bit and I started researching with small-scale fisheries along the Pacific coast of Costa Rica. And throughout all that time, I just wanted to improve the relationship or study the relationship between human beings and nature and how we all use nature. So I went to go get a PhD at the University of British Columbia between 2015. I just finished the PhD, and there I studied how climate change might be affecting fisheries in the Eastern Tropical Pacific. Now I am doing a postdoc with my advisor, who was William Chung at the Changing Oceans Research Unit, and with Sonia Bejarano at the ZMT. And for both, I'm looking at how climate change might affect reef fish in the Eastern Tropical Pacific and at the global level. So that is what I'm sort of focusing on now. Today, I am going to be talking a little bit about the Aerobic Growth Index, which is a method that William and I developed throughout my PhD. And then I'm going to show you how I applied it to fisheries in the Eastern Tropical Pacific to study the impacts that warming and oxygen limitation or deoxygenation may have on them. So I already touched on this a little bit, but I would like to start by acknowledging that this is the research that I'm going to be presenting today is not, I didn't do it on my own. It's the result of a collaboration throughout my PhD with my then advisor, William Chung. And we also worked very closely with Thomas Fröhlicher and Anne Murray at the University of Bern in Switzerland, and also with many other researchers and students at the Changing Ocean Research Unit and the Institute of the Oceans of Fishery at UBC and beyond. So we know that the ocean has absorbed over 28% of anthropogenic greenhouse gases and over 93% of excess heat. And so while this has made it so that the ocean plays a big role in stabilizing the climate and mitigating climate change a little bit, it's also changed the ocean environment. It's changed marine habitats. And the effects of climate change are widespread. Changes in temperature, in oxygen, in ocean pH, in sea level can be seen in all of the world's marine regions. And warming and deoxygenation are some of the most important climate stressors, especially when you're thinking about biological impacts. So warming sort of causes deoxygenation in the open ocean by reducing the amount of oxygen that can be dissolved in water. It increases stratification, so it reduces the mixing between the highly oxygenated surface waters and the low oxygen midwater, so you get less transport of oxygen and renewal of oxygen at mid layers. And at the same time, you get an increased demand because our biological organism will increase their respiration rates and their demand for oxygen. This has made it so that between 1970 and 2010, the ocean has already lost between 0.5 and 3.3% of its oxygen content. And under a worst case scenario, we would expect that the ocean would lose an additional up to 4% of its oxygen content by the end of century. And the oxygen or deoxygenation isn't spread homogeneously throughout the entire ocean. We have these oxygen minimum zones in what you call the shadow areas of the Eastern Pacific Oceans, especially in the tropical regions. And this is where sluggish ventilation reaches very high respiration rates. So you're not getting lots of supply of oxygen, and there are more organisms extracting the oxygen from the waters. And this is where we can get oxygen levels as low as 0.05, 0.08 moles per cubic meter. And the deoxygenation or oxygen loss in these areas is occurring a lot faster, 10 times faster than the global deoxygenation rate. And it's estimated to be at about 4% the oxygen content per decade. This has made it so that between 1960 and 2010, the volume of these oxygen minimum zones has quadrupled. And right now they represent up to 8% of the area within the ocean. As you saw on the previous slide, the Eastern Tropical Pacific is home to very large oxygen minimum zones. And not only do concentrations get below the estimated threshold of 0.05 to 0.08 moles per cubic meter, but they also can have functionally anoxic cores. And these oxygen minimum zones can have the potential to affect coastal biodiversity and coastal fisheries because they shoal up to around 40, 50 meters below the surface, where waters are quite warm and where we would expect to have a high marine biodiversity and where lots of fisheries act. These oxygen minimum zones have been expanding, and so what can happen is that it can squeeze species that are hypoxia intolerant, which are most coastal species, and they can squeeze them closer to the coast where they would be more vulnerable to coastal hypoxic events or to fisheries or to pollution. And just as an aside, yes, I started to concentrate on oxygen minimums, ocean deoxygenation and warming as climate impacts because I think that it's incredibly interesting. But also, when I went up to do my PhD, I decided that I wanted to study the impacts of climate change on the Eastern Tropical Pacific because I grew up here. I wanted to sort of study this region and see how I could learn more about how climate change would be affecting fisheries in the region. But then I realized quickly that deoxygenation, these expanding oxygen minimum zones, were going to be shaping the main climate impacts on fisheries resources. And that is where I started to shift my attention to ocean deoxygenation and warming, which luckily was also a main interest of my advisor at the time, William Chung. The Eastern Tropical Pacific, I also think, is a really interesting place to study climate change impacts because of its very strong latitudinal temperature gradients. To the north and to the south of the region, it has strong upwelling produced by the California current, by the Humboldt current, and by equatorial upwelling. So this brings cold waters with low oxygen to the surface, and it encloses a warm pool that exists around Central America. So the very unique temperature gradient along the Eastern Tropical Pacific, where there are colder waters actually close to the equator, means it so that species are projected to shift in directions and in a way that are very unique to the region. Species along Central America are actually shifting towards the equator, towards the cooler water that is brought up by these upwelling events that tends to have temperatures that are more favorable to them. And another thing that I found was that species are projected to shift towards shallower waters instead of towards the deeper, cooler waters that are generally seen. And this is because of the expansion of the oxygen minimum zones. And we see that in countries where, like in exclusive economic zones, where the oxygen minimum zone is already very, very close to the surface, such as Nicaragua and Costa Rica, that is where we're going to see the smaller depth shifts because there's actually very little, they don't have much more to go. They're so close to the surface already. And all of these changes in the physical and the chemical conditions in the ocean can propagate through the organism, the population, the ecosystem level to affect fisheries. And this can then affect the way humans depend on fisheries, whether it's for cultural purposes. Target species might change, culturally important species may no longer be available. This can affect how we depend on fisheries for food. It can affect food security in coastal communities. And it also can affect how important or the way that fisheries support coastal livelihoods. So let's begin by talking about the mechanism by which warming can affect the actual organism. And that is because warming increases the metabolic oxygen demand of marine ectothermic organisms. And so as temperatures warm, oxygen demand increases while maybe oxygen supply in the environment remains the same or may even decrease. And this is how combined warming and deoxygenation affect the fitness of marine ectothermic organisms by compressing their aerobic scope. Now, marine fish, they need oxygen for a whole bunch of processes that they need to survive. They need it for survival, they need it to feed, to move, to grow, to reproduce. And so while warming increases the amount of oxygen that they need to do all these things, then the oxygen supply might decline, reducing the amount of oxygen available and causing the organism to have to make trade-offs. Well, do I allocate the oxygen available to growth or to survival or to reproduction? And then that can affect the fitness of the organism and eventually the population size. But not all species are affected in the same way by warming. Now, some species, which tend to be adapted to warmer waters, actually have either physiological or behavioral adaptations that allows them to withstand and to have they tolerate the warmer waters and deoxygenation, while the colder species tend to have higher oxygen demand. Like the species that are adapted to colder waters tend to have higher oxygen demands and therefore be a lot more sensitive to deoxygenation. So we know that warming drives changes in catch composition, but I wanted to explore what the role of oxygen limitation might be as a mechanism causing those changes in catch composition. So we would expect that at the cool temperatures, that cool adapted species with higher oxygen demands would dominate the catch. But then as temperatures warm, their abundance would decline, the abundance of the warm adaptive species would increase, and therefore the catch would then be adapted to would be dominated by warm adapted species with low oxygen demands. And so motivated by an understanding of the potential biological impacts of warming and deoxygenation on fisheries in the Eastern Tropical Pacific, and trying to study those biological impacts in an area where there's high data deficiency, then we developed the Aerobic Growth Index, which is a metabolic index which can be applied to with very few data. It has very few data requirements, and it's based on how species growth and metabolism relates to their environment. It's essentially an ecophysiological indicator of habitat viability for marine fish and invertebrates. It's a ratio of oxygen supply in the environment and oxygen demanded by the species. So in that way, if oxygen supply is higher than the oxygen than the species demands, then the habitat is viable. Warming can affect habitat viability by increasing the oxygen demand above supply, and deoxygenation can affect habitat viability by lowering supply below the oxygen demanded by the species. And so in this way, this index is able to project where habitats might be lost along the warm range of a species distribution, or along oxygen minimum zones, or in dead zones, or during coastal hypoxic events where deoxygenation would be the main driver of habitat loss. It cannot project habitat expansions because this version of the Aerobic Growth Index does not yet include the mechanism that defines species distributions at the cooler range of their distribution. So we then set out to validate the Aerobic Growth Index using a previously published metabolic index. And we were able to do this, and we had such confidence in the previously published metabolic index because it's based on physiological lab experiments determining species oxygen thresholds at different temperatures. So this is highly reliable information. But the problem with it is that it's also not available for most tropical species. So this is where the Aerobic Growth Index comes in. It's based on the von Bertalanffy model, and its main input parameter is the weight at infinity. So it can be applied to thousands of marine species for which this information is available. We were able to compare them because they're basically the same thing. They're a ratio of oxygen supply in the environment to the oxygen that's demanded by the species, but the actual interpretation of them is a little bit different because the metabolic index uses oxygen demand at the onset of anaerobic metabolism, but the aerobic growth index uses the oxygen demand at no growth. So the way that we would interpret the results is slightly different. And I'm not expecting you to be able to apply the method or to fully study the method in this slide or the equation. The only thing that I wanted to show you here is that the input parameters here they're not very many. So we need the asymptotic species weight, which is from the von Bertalanffy growth equation. And then we use the Arrhenius equation and Arrhenius constants that were derived by a meta-analysis for fish. So that information is constant across species and it's available for all, it's a constant parameter. You don't need to have that additional information for your study species. And then you would need information on the oxygen and the temperature throughout your species distribution. So you would also need some information on how they are distributed. But with the advent of GBIF and OBIS, there's a lot of really great information on species occurrences out there. So we applied the aerobic growth index and we compared it to the metabolic index. Here we're projecting the habitat loss for 3 different species. Where you see red in the maps, that is where both methods project habitat loss in the same areas. And this is comparing with 2 different versions of the metabolic index. So we see that the aerobic growth index actually is able to project the habitat loss in both the percentage of the original baseline species distribution and the spatial patterns of habitat loss quite well. We also wanted to make sure that the aerobic growth index also is able to work with invertebrates. So we looked at we compared habitat loss using both indices for these 3 invertebrate species and found also that both indices project habitat losses in very similar places. So with this information and this validation, we were confident with the fact that we can apply the aerobic growth index to study the impacts of warming, deoxygenation, or oxygen limitation, depending on your habitat, on marine fishery species. And that is exactly what we did with the next step, applying it to pelagic fisheries in the Eastern Tropical Pacific, hypothesizing that changing ocean temperatures and oxygen levels are playing an important role in shaping the catch composition of pelagic fisheries in the region. So first, to be able to understand the results, we wanted to have a brief understanding of how temperature and oxygen changes in the Eastern Tropical Pacific. Because it's pelagic fisheries, we were looking at the surface level, where we found that the average annual temperature ranged between 23 and 29 degrees Celsius. The average annual oxygen concentration between 0.2 and 0.22 moles per cubic meter. And we found that in the north and in the south exclusive economic zones, this is where you find the lowest temperatures and the lowest oxygen concentrations. And this is probably due to the upwelling that brings the cooler, less oxygenated waters to the surface. And we scaled up the aerobic growth index to be able to see how to study the actual catch composition by creating a catch-based index. The first one that we generated is the mean aerobic growth index of the catch, which would be an indicator of the physiological performance of the average species within that catch. And it's essentially the aerobic growth index for each species weighted by their catch. And it allows us to capture species responses to warming. And in this case, I expect the latitudinal temperature gradient that you see along the eastern tropical Pacific. And then we also use the mean oxygen demand of the catch, which is an indicator of how sensitive the exploited species in the catches are to oxygen limitation. The higher the mean oxygen demand of the catch, the more sensitive we would expect that particular catch composition to be, because it would need more oxygen to remain the same. And this index is also catch-based, and we average the oxygen demand estimated at the species' temperature preferences, and we weight that by the amount of catch. And what it allows us to do, if you remember the slide that I showed you previously about the different species, either the warm adapted species or the cold adapted species, being more or less sensitive to deoxygenation according to how much oxygen they demand. So this index allows us exactly to see that. It allows us to see how vulnerable species would be to oxygen limitation within the catch according to how well adapted they are, how much oxygen that they would be demanding. So we found that when applying these indices to the pelagic fisheries in the region between 1970 and 2016, we find that in the Ecuador and the Galapagos EEZs, there is actually a threshold, a breakpoint within the relationship between these two indices and oxygen. And one would expect oxygen to act as a threshold that would separate different catch compositions of the fishery. So here we see that at low oxygens, the catch is mainly dominated by species with lower oxygen demands. And then after a threshold, then the catch composition starts to change and being dominated increasingly by species with higher oxygen demands. And so this is related to upwelling, where lower oxygen levels are related to species with lower oxygen demands. The breakpoint is at around 0.2021 oxygen partial pressure, and the AGI seems to remain constant until that threshold and then starts to decline. And why is that? And that is just because the species composition is changing. So as species demand more oxygen, there is a lower space or a lower surplus available for each one for those species. Another, I think, interesting thing that I found when studying both of these indices is that I didn't find many long-term trends, but I did find that the indices vary with El Nino, and the the colored lines represent the mean oxygen demand of the catch and the aerobic growth index of the catch for each one of the 3 areas with similar environmental conditions. And the black index represents the standardized oceanic Nino index. So it represents the El Nino and La Nina events. And I thought that it was very interesting to see that, well, higher index levels mean that you get stronger El Niños, which is when you have warmer temperatures, but also a lot more oxygen in the water. And so I thought that it was interesting to see that the mean oxygen demand of the catch increases when we get strong El Nino events. In contrast, the aerobic growth index of the catch seems to have an opposite relationship with the El Nino and La Nina events. So we then applied both of these indices to study a demersal community along an oxygen minimum zone. And there we thought that, hypoxia tolerance, or that how sensitive species are to very low oxygen levels, would be the main factor that drives the changes in both of these indices. And because the low oxygen minimum zone that we studied was along the Pacific coast of Costa Rica, and because we don't have really long time series there, we got a little bit creative and started to use natural experiments created by both the depth gradient and El Nino and La Nina. So we were sampling the shallow end of the oxygen minimums, where there are very, very strong temperature and oxygen gradients that would structure species communities. And in this area is also where you can see how the oxygen minimum zone expand and contract, which they do across seasons, they do between El Nino and La Nina. In El Nino conditions, upwelling intensity decreases. So that is when we get warmer waters, and the oxygen minimum zone at the thermocline is pushed deeper. And so at a given depth, we have higher oxygen levels. During La Nina, the upwelling intensifies, waters cool, the thermocline gets closer to the surface. So the same with oxygen minimum zone, it gets closer to the surface. So at a given depth level, we would find lower oxygen levels. And I think one of the most interesting things that we found was that species depth distributions sort of change with El Nino and La Nina. In El Nino conditions, where there's more oxygen, species depth distributions are deeper than during La Nina, where there's lower oxygen, and so species actually go towards shallower water, searching that higher oxygen levels. And during La Nina, the depth distributions of all species were shallower than during neutral or Nino conditions. And it was interesting because I actually was on these sampling campaigns. I was on the boat. I was talking to the fishermen, and it was incredibly noticeable, the changes in distribution on board. Also during La Nina, we had a lot more jellyfish events. And talking to the fishermen, they said that yes, this is an incredibly common pattern, that species shift shallower or deeper according to how cold the water is. And so we expect that if we're seeing this with El Nino and La Nina, that as oxygen minimum zones expand closer to the coastline with climate change, as has been shown to happen in the California current, that species would also be shifting towards shallower waters, and they would also they would be squeezed closer to the coastline. So when we compared both indices with temperature and oxygen, we also found that an oxygen threshold separates both communities. And that oxygen threshold was 0.03 to 0.09 oxygen partial pressure, which is a lot lower than for the pelagic community, but it's something that we would expect given the conditions that these species are adapted to. And we expect that this threshold is separating the community within the oxygen minimum zone and the one in the oxygen limited zone. And another thing to notice is that because we didn't have in situ oxygen information, we had to use hindcast, and there is an incredible variability in the oxygen information for each model, which really shows in differences in the results calculated according to which oxygen data that we use. And so this really highlights the need that we have to start establishing oxygen monitoring programs in the region. So I would like to leave you with 3 key messages today. I would like to for you to go home thinking that the Aerobic Growth Index can be used to study the impacts of warming and deoxygenation on marine ectothermic organisms. And we have shown that temperature and oxygen limitation shape pelagic fisheries catch composition within the Eastern Tropical Pacific, and that oxygen supplies should mainly shape the composition of the demersal community along oxygen minimum zone. And so we would expect that if temperature and oxygen are playing such a strong role in shaping the composition of these fisheries and communities right now, then any changes in these conditions would then also lead to changes within the fisheries and within demersal fauna. And I think that the Aerobic Growth Index can be applied to many more things, can be used to answer many questions. And some of the ones that I'm working on right now with the oxygen team is looking at compound extremes, so extreme warming events, extreme deoxygenation events, when they happen together, how that might affect marine species, and applying the Aerobic Growth Index at a 3-dimensional level instead of 2-dimensional, so that we can actually see habitat compression at the depth level rather than just changes in habitat area. Then I think it's important to apply it to study biological impacts on global fisheries. And I'm now applying it to reef fishes, which have a lot less information than available for them than the global fishery species. And I am also in the finishing touches of modifying the Aerobic Growth Index so that it can now project habitat expansions in addition to the habitat losses. So it can project shifting habitat areas in total, and not just how much we're losing of them. And I thank you very much for your attention, and I would love to discuss any questions you may have.
Thanks, Taylor, for your excellent presentation. It's very nice to see the more details your work, is very complex, in my opinion, to work. Well, people, now I will open the one space for the questions and I change again the language. Pessoal, então vamos, quem tivesse vontade agora de fazer alguma pergunta para ela, por favor escreva suas perguntas aqui para a gente conversar com ela um pouco mais sobre os resultados que ela apresentou o dia de hoje. Well, when people start the question, I have very interesting in your topic and you understand more about the climatic change effects, for example, the distribution for the species. Mm-hmm. And the future scenarios is the temperature increasing more, and this one reflecting in the oxygen concentration. But in many cases, you have the shallow reef is more affecting, but the mesophoric reef probably not. And what are you expecting about this change in the distribution range in the species, for example?
Mm-hmm. Yes, I think that for reef species and for pelagic species, I would expect that the warming increase would be the main driver. And so we're able to show how just warming can affect the habitat areas for them, and through the mechanism of oxygen limitation. So even if just warming and not deoxygenation is affecting them, then this method is able to capture that. And I have some initial projections of how reef fish habitat might change in the future. And I guess it's not surprising that under the worst case scenario, up to 80% of the reef species that I'm studying, which is they're like 1,000 species, they lose their entire habitat. And so considering the projections for actual corals, this doesn't seem it doesn't seem too surprising. But I do think that ocean deoxygenation and coastal hypoxia are also going to be are also affecting reef fish and reefs in general, but we just don't have the information to detect those coastal hypoxic events. And the models that we're using, they don't project that. And so again, this is where we really need to start measuring oxygen to start detecting those hypoxic events and seeing how they might affect species and how we can prevent them.
Yeah, I agree totally. This is very important metric. Well, this is another curiosity. Probably you talking about in your presentation, but the tropical Eastern Pacific have two different outwelling. One outwelling in the north, more near for the Mexico, and another more near in the Ecuador, Peru, Colombia, and this is very strong outwelling, and probably it is very proportion many nutrients and many oxygen in this case. And but my question is more in terms of biogeography, because this is one limit for different tropical species, and probably in the more hot areas you lost more oxygen and probably you have more extinction. But this is possible, thinking about the more extinction in tropical areas in comparison in this specific outwelling or buffer the oxygen you have?
Yes, definitely. It's going, I mean, there are different ways that it could go, and the projections for upwelling areas are not that great. There's a lot of information. I mean, they're getting better, and with longer and longer historical trends, we're understanding a little bit more how they've changed and how they could change. But so if they intensify, then they could intensify to the point where you're creating these hypoxic areas. And so your species are losing habitat because they're not able to survive in these areas with such little oxygen. But if the intensity is not as strong, then species might benefit from the cooler temperatures and the high primary productivity. So it's going to depend what model you use, I guess, to project the upwelling events and exactly how intense how more intense the upwelling events get. But also, as oxygen minimum zones expand, then the water that the upwelling events are going to be drawing on are more and more hypoxic. And so that also increases the probability of bringing really low oxygen water to the surface or right below the surface and, causing habitat loss and affecting species. So there is a lot of uncertainty with what might happen and how upwelling areas might benefit or not fish in the future.
Nice. Well, this is my last question. Is more about the method. Is for example, is the oxygen measure is very variable and you use it one specific online repository, and you suggested started increasing the measures in the field work. But what is the advantage you have when you have more nature about this field work?
Sorry, I just want to make sure that I understood correctly.
I can repeat. Mm-hmm. My question is, what is the advantage when you measure the oxygen? This is very variable. You can measure now, and 5 minutes after is probably changed. Mm-hmm. And what is the benefits you increase? You include this measuring the monitoring program, for example.
Mm-hmm. So I think it all depends on the questions that you want to answer. If you want to just look at average changes in habitat area by the end of century with then just using the Earth system models that we have available, that's okay. But if you want to look at extreme deoxygenation events where you get these dead zones that, as you say, can be very, very variable and can happen during the day or night, change even across different times of day, then that is where you're going to need the more detailed variable oxygen information if you want to see how habitat area changes with oxygen levels on the reef fish, just at an hourly or daily or just at a more specific time, which I mean, it would be really, really useful and really interesting to see just pure biological or ecological curiosity, but also it could help you manage or preserve those reef systems a little bit better as well. I don't hear.
Sorry. I closed because many people talking here. Pessoal, alguma outra pergunta para Tayler do dia de hoje que quisessem agregar aqui? Senão eu vou dar por terminado no CEBIMário de hoje. Eu acho que é um tema bastante complexo. Eu sugiro muito ler o paper dela, está publicado, para entender um pouco mais sobre o que ela faz e ela que tratou de explicar o dia de hoje aqui. Well, Tayler, thank you again for participating in this initiative for the CEBIMar. It's very nice. You understand more about your work and maybe continue to collaborate for different people in Brazil and the other people probably watch this video.
Thank you. Obrigada. And if anyone would like to send me an email with a question or, question about how to apply the method or just anything, just please reach out and I will be more than happy to get back to you. Thank you very much.
Thank you so much. Bom, pessoal, então vou dar por terminado no CEBIMário de hoje e agradeço a todos que nos assistiram presencialmente e quem irá nos assistir em algum momento essa gravação. Aproveito então para mencionar que a gente vai continuar então com a temática de mudança climática. A semana que vem vamos falar então vamos escutar uma palestra sobre mudança climática em Galápagos especificamente, da doutora Margarita Brown, e espero poder que vocês participem desse momento ou dessa discussão com a professora Margarita Brown. E aproveito de novo a oportunidade para mencionar que se vocês querem trazer seus trabalhos e apresentar nesse espaço aqui, é só encaminhar um e-mail para mim e com muito prazer a gente agenda um espaço para vocês trazerem aqui. Thanks, Tayler, and see you in another opportunity. Bye bye para todos aí. Tchau.
Thank you. Tchau.